Hello everyone. Today let's see what is gene therapy. Definition: it is an intracellular delivery of genes to generate a therapeutic effect by correcting an existing abnormality. What are the types of gene therapy? There are two types: somatic gene therapy and germline therapy. Somatic gene therapy is the one under trial, and germline therapy is considered to be unethical. What is the whole procedure of gene therapy all about? Let's see one by one. First, isolate the healthy gene. Second, incorporation of this gene on a carrier or a vector as an expression cassette. A vector is considered, and this healthy gene is introduced into the wild vector genomic sequence and as a cassette, as an expression cassette. And lastly, delivering this vector to the target cells, which is the deceased host cells. Into that, the healthy gene, which is incorporated into the vector, is introduced. How the gene carrying the vectors are introduced. Now we have to know how this is introduced into the host. Three methods are there: ex vivo strategy, in vivo strategy, and in situ strategy. Coming to ex vivo strategy. Looking at the picture, first you have to take the cells from the patient, and the cells with the defective gene now is outside the body. Thirdly, the retrovirus carrying the normal gene is considered, which is the vector now, and it is cassetted with the normal gene, and that is ready and all set. Now we have to introduce this retrovirus into the patient's cell. So the normal gene is introduced into the patient cells by the virus. The virus itself will introduce the normal gene into the patient cell because the retrovirus is infective. Fifth, the patient cell is now containing the normal gene, and the patient cell is introduced back into the patient. This is a very rough idea about the ex vivo strategy. What are the vectors used in gene therapy? Viral vectors, non-viral vectors, and physical vectors are there. So coming to the viral vectors, retrovirus and adenovirus, adeno-associated virus, and herpes simplex virus. These are considered to be two categories. We will discuss it later. Next is the non-viral vectors, which are plasmids and liposomes. Thirdly, the physical methods. There are three physical methods: gene gun method, electroporation method, and microinjection method. Coming to the retrovirus as a viral vector, this is a very important topic and commonly asked questions in exams. The most commonly used retrovirus is MMLV. We have to include the advantages and disadvantages of it, and that completes the answer to the question. Gene transfer by retroviral vector. Here you can find the retrovirus, which is of the wild type or the original type with its RNA sequence: LTR, GAG, POL, ENV, LTR. Next, during the gene transfer, delete the GAG, POL, and ENV genes and place, instead of the set of three genes, a normal human gene, which is the gene of interest. Defective virus is put into packaging cells which provide the viral code. Defective virus in the sense it has been now modified, introducing a human gene into it, right? So now we find the viral particles with the protein coat. The protein coat is necessary for the virus to be infective. These viruses are infective and can enter into human cells. Infective in the sense it can enter into the human cells for its purpose as a vector. It will not replicate and it will not produce any disease. Look at the picture here. The retrovirus being infective is entering into the human cell cytoplasm via receptor-mediated process. The modified RNA sequence with the human cassette, which is a blue colored one, human healthy gene cassette, is introduced into the cytoplasm, and it has to now enter the nucleus. But before that, the RNA sequence of the retrovirus has to be converted to a DNA sequence. So the reverse transcriptase of the virus will copy the RNA to the virus DNA and then confer the changes so that it can make changes in the host DNA, right? So once the virus DNA is formed by the reverse transcriptase, it can integrate into the host DNA inside the nucleus. Coming to adenovirus, it is a DNA virus. Retrovirus was an RNA virus, right? This is a DNA virus. There are two types, type 2 and 5. Viral DNA becomes epichromosomal. This is important. It doesn't enter into the nucleus. It remains as a different unit outside the nucleus. Advantages are it is produced in high titers and it can infect wide variety of cells. Disadvantages is the expression is a transient phenomenon. Okay, it is transient, it is not long lasting. Now coming to adeno-associated virus, it is a DNA virus. There is no pathogenic effect, none. It is used in hemophilia treatment and site specific on chromosome number 19. Herpes simplex virus. It is a DNA virus and it infects neurons. Advantages are it can insert large DNA sizes and it's produced in high titers and it produces long-term CNS gene expression. We can classify the viral vectors basically into integrating viral vector and non-integrating viral vector. Integrating viral vectors are the retrovirus and adeno-associated virus. Non-integrating viral vectors are adenovirus and herpes simplex virus. Integrating viral vector in the sense the vector can integrate into the host DNA, while non-integrating viral vector remains as an epichromosome. Now let's learn non-viral vectors. First is the plasmids. Plasmids are very important in the field of recombinant DNA technology. Look at the picture here. Plasmid with the gene of interest is isolated from the vector and it is directly injected into the organism, and this is the in vivo technique. The ex vivo technique goes like the plasmids with the gene of interest is considered in vitro and that is directly introduced into the organism. What are the features of plasmids? It can accept DNA of 6 to 10 kilobase pairs. It is mainly used in DNA vaccination and efficiency of uptake by the human cells is less than 1%. It does not integrate with host DNA. Coming to the other set of non-viral vectors, which are liposomes. The plasmid carrying normal gene is first inserted into the liposome and you can find the liposome plasmid complex here. And the plasmid liposome complex enters the host cell by fusion of the membranes and it will not enter into the nucleus. This is very particular. It will not enter into the nucleus but will remain as an epichromosome outside the nucleus. So it is staying as a different unit and it will start expressing the normal gene in it. What are the advantages? It can carry the human gene of big size and it do not replicate, only weak immune responses produced. These are the advantages of liposomes. Disadvantages, it is easily destroyed and the gene transfer efficiency is also less. Now coming to the physical methods, gene gun method, electroporation method and microinjection method. Gene gun method. The tungsten particles are coated with plasmid DNA which is carrying the healthy gene of interest and it is accelerated by helium pressure discharge. Advantages are it is a very quick procedure and it directly targets the tissue. Directly the considered gene of interest is introduced into the target tissue. What are the disadvantages? Cellular damage can happen there, and the gene expression is a very transient one. It is not long lasting. Next one is electroporation. The cells are exposed to high electric field strength in the presence of gene. That's all you have to know. The cells are exposed to high electric field strength. Microinjection method. It is the direct injection of the foreign DNA under consideration, or the healthy DNA, into the host nucleus. There are different success stories of gene therapy. Let's just brush up through these. You can find it as a table in your textbook. Let's see 4 or 5 of them. Firstly, severe combined immunodeficiency disease, or SCID case. You can find the gene transferred is adenosine deaminase enzyme and the vector used is a retrovirus. DMD, or Duchenne muscular dystrophy case, you can find dystrophin gene is the gene considered and retrovirus is the vector. Third, cystic fibrosis CF. In that case, the gene considered is a CFTR gene and the virus considered or the vector used is adenovirus. Next, you can look into the cancer. In that case, the gene used is activation of P53. P53, you might have studied earlier that it is a tumor suppressor gene. So we introduce the P53 gene into a cancer cell. And the vector used is a non-viral vector, which is liposome. What are the limitations of gene therapy? Lack of an ideal vector is a problem. There is a lack of targeting ability in non-viral vectors. We have seen that in non-viral vectors, they remain as epichromosomes. Gene therapy is a short-lived one. It is producing inconsistent results. It produces immune responses and multi-gene disorders, and it is expensive. Thank you for watching.